Hello, everybody. So it's my absolute pleasure to introduce Thomas Vidback from Brandeis University, and he's going to be talking to us about arts and craft inspired self-assembly. So, Thomas, it's over to you.
Thank you very much. Thanks for inviting me to come and give a talk. Uh, as Adil said, I'm Thomas. I'm from Brandeis. I'm a research scientist there. Um, and today I want to tell you about some fun stuff we've been doing, thinking about these kinds of triangulated structures and how to cut and paste them together and realize them in self-assembly systems. And so our lab, we're really interested in the process of self-assembly. And for those who don't think about it often, it's this field where uh, you think about how local interactions between particles can lead to some kind of larger scale emergent structures. And I always love this image here on on the left, which are these really large kind of salt crystals in a cave in Mexico, where just kind of atomic scale interactions give rise to these beautiful ordered structures. And just for reference, these little orange blobs are actually people going inside, right? And so you get something that's from the atomistic scale up to many meters in length, right? And maybe you've done this type of self-assembly, this type of crystalline self-assembly before when you were young. You've taken string and maybe put it in sugar solution to make rock candy. And so inspired by this type of natural phenomena, people in the soft matter field have been interested in trying to emulate this type of kind of ordered arrangement and local interactions. And one way that people have been really interested in doing this is on the colloidal scale. So here are some examples of particularly colloidal particles that have been uh, grafted DNA strands onto them, so you can play around a bit more with the way they interact, but you can get different types of crystalline structures here, either by changing the specificity of a particle. You can make like a red and a blue particle that interact, or David Pine has some really beautiful work where he makes these tetrahedral clusters that can interact with each other and form this ordered diamond lattice. And so it's nice that you can make these kind of bulk crystalline materials. But if you look at biology and you ask what kind of structures do we see there, you find a really diverse field of types of things, right? You get all kinds of different shapes. They all have very different function. And one thing that's really key about them is that they aren't bulk crystalline states, right? Often they're finite in size. They have defined curvature across their surfaces. And so I just want to point out a couple of these here. There's examples like the microtubule or the viral capsid where they have some kind of curved surface to them. And there's also objects like the ribosome, which is made out of a bunch of unique biomolecules, which has a lot of complex function. And so I want to focus down in this kind of biologically inspired realm of structures to structures that are curved membranes, right? So I had already talked about viral capsid and microtubules. These are analogs of either spherical shells or cylindrical shells, but they're also more exotic types of things in biology, like the torovirus. I have a question mark here on the toroid shape because if you look very closely, these aren't fully closed, but you can think about them as being almost like a donut or a spirochete, which has this interesting kind of helical tubule membrane. And so one question you can have is, if you want to make these kinds of more interesting curved membranous structures in a synthetic system, right? How are you going to go about doing this? And we can look at some examples of what have been done in the past. On the left, there's some examples of kind of colloidal attempts at making filaments, right? And so these ones work. You can have colloids assemble into filamentous structures, but if you wanted to try and make a hollow cylinder out of colloids, it's really difficult because often these types of particles are either isotropic or on this bottom left, they're Janus, and so they often tend to pack very densely, and so it's hard to make kind of voids in them. And then in the middle and the right are examples of chemically synthesized fullerene structures, and these are able to make really cool kinds of curved surfaces. Here are examples of cross sections of carbon nanotubes. And on the right here, we can see examples of these curved helical structures or these double donuts. But these structures are often very tricky to make. You have to have very particular synthesis protocols, and they're often very polymorphic. And so what I want to try and do today is think about how can we try and design from the bottom up a self-assembly system where we can really try and make these types of curved geometries and make them very precisely. And so to break this down, In the first part of my talk, I want to talk about kind of a simple curved surface, a cylindrical sheet. And so we're going to ask, how can we try and design this, and how can we specify this as accurately as we can? And then at the end, I'll go into thinking about ways to make these kind of more complicated curved surfaces, like the spirochete. And so to think about, how am I going to go about trying to create a self-assembly system? We need to think about ways in which, or what we do is we think about, look at biology and see how has it tried to make these kinds of finite curved structures. And there's two kind of paradigms that I want to point out for this. One of them is something called addressable assembly. This isn't particular for a curved membrane, but it's an interesting way that biology organizes material. In the case of the ribosome, this is a structure that's made up of a bunch of unique biomolecular particles, and each one has a home in the final structure. And so if you have all of the ingredients, this will be the only kind of assembled structure that it will make. Each particle in here is addressed in a unique way. Whereas in the case of a viral capsid, here you're trying to make a curved surface. And in this case, the proteins, when they come together, this example here is actually made out of a single protein subunit. And when these things come together, they bind, but they bind in particular locations on the subunit, and they bind at a particular angle. And so as these things come together, they will accumulate curvature across the surface and then eventually close on themselves. And this form of assembly, where you use a kind of encoded curvature between assembling subunits, can be a very economical way of creating structures without having to address all of the different components in your system. And so I'm going to talk a little bit about how we can put these two types of things into effect. But before we can do that, we need to know, like, what are we trying to make? And so we're going to be thinking about how to make this type of cylindrical structure. And the way we're going to do it is we're going to try and think about how to make it out of triangles. Triangles are a nice choice because you can typically triangulate any types of surfaces you want. So if you can find a way to encode these interactions into triangular subunits, maybe you can make whatever types of surfaces you want to. And we have an additional constraint here that we're considering equilateral triangles. And one way to think about how you can take triangles and turn it into this sort of cylinder shape here is, if we imagine just a triangulated sheet, we can pick 2 points, 2 vertices on this structure, and we can ask that these 2 points are periodic with each other, right? And so when you make this kind of request, you can draw a line between them. This will end up being the circumference of your tubule, and these dotted lines here will be these periodic boundary conditions. Just to visualize this a bit better, here's a triangulated sheet, and hopefully you can see these 2 cyan dots. I guess I can use this laser pointer, right? So we can take these 2 cyan dots here, and we can close this sheet up onto itself. And in the process of bringing these 2 points together, we've made this curved surface. And the nice thing about this is that locally everywhere on this structure, it looks the same. And so you can actually make this type of curved particle, this curved cylindrical sheet, out of a single type of particle. Just for some nomenclature, this is a discrete thing, right? There's a finite number of triangles in the circumference of this object. And so I just want a way of labeling these types of shapes. And so what you can do is, along this circumference direction, you can ask, what's the shortest path you can take to go from one point to the other, walking along one of these lattice directions. And so we're just going to call these M and N as a way to distinguish the type of tubes that we're making, these types of cylinders. And this one here is an example of a 10-4. If you walk along this white direction, you take 10 steps, and then you'd have to take 4 back down in this red. And the nice thing about this type of construction is that just by changing these kind of M and N numbers, you can target a variety of different cylindrical shapes, right? You can either change the diameter, you can change the helicity of them. And in an ideal world, you can do this with a single type of subunit, because any particular triangle you look at here is going to be the same as any other one. And so let's see how we can try and make a self-assembling subunit that creates this type of structure. And so this was a problem that was really figured out by two of my colleagues, Douglas Hall from UMass Amherst and Daichi Hayakawa, who's a graduate student at Brandeis, and now he's a postdoc at Caltech. And so we can ask, how are we going to try and make this type of cylinder? It's a 10-0 tubule, so it's 10 triangles in its circumference. And what we need to do is we need to create a triangular subunit that has 3 unique sides. And the reason we need to have these 3 sides be distinguishable from each other, since in this cylinder we've suggestedly already colored it this way, you can see that there's these 3 primary lattice directions that wrap around this cylindrical surface. And if we look at this 3D model of this cylinder, we can actually ask what the kind of angle between adjacent triangles is. And what we see is that in general, along these 3 different lattice directions, you have different curvatures. And so these triangular faces have different angles between them. And so what we're going to think about is trying to encode that angle between adjacent triangles by programming in a binding angle by taking the face of a triangle and tilting it a little bit. And so depending on how you tilt or bevel this angle, when 2 triangles come together, they'll come together at a specified angle. And the last thing that we'll need to do to encode into our subunit is to make sure that these interactions are specific. And what we mean by that is that this red side will only bind to red, blue to blue, and white to white. And we're going to end up doing this with DNA, which I'll show you. And so these are kind of the requirements that we have for these self-assembling particles to make this kind of sheet. You need to have well-specified binding sites. So you have to have control over the valence of your particle. You have to control the binding angle, and you also have to make sure that these things are have specific interactions. And this kind of list of requirements is hard to do with kind of standard colloidal particles. And so we turn to a different type of tool to make these types of structures. And so what we do is we use something called DNA origami. DNA is this nice biomolecule that forms double helices. When a double helix forms, it actually becomes quite rigid. But one of the other really nice parts about using DNA is that it has this thing of base specificity, right? It has 4 bases in it. There's these G's, C's, T's, and A's. G always binds to C, T always binds to A. This is this Watson-Crick base pairing. And what that means is that if I have 2 sequences of DNA with complementary sequences, we can be pretty sure that they're going to bind to each other. But the other nice thing about this kind of base sequence specificity is that you can also do tricks with it, where if I had a really long strand of DNA, say this black strand, and then I had a shorter strand of DNA that had complementary sequences to disparate parts of this black strand, you can actually pinch this larger strand together locally. And so what this means is, and this kind of pinching of non-local regions of other DNA strands is the core of DNA origami, where you take a large single-stranded plasma loop, you find 2 regions of it that you want to bind. You can design and order a small oligo that will locally pinch this together. And if you design a right set of staples, you can create kind of arbitrary 2-dimensional or 3-dimensional shapes using this method. And so we're going to try and use this technology to make this 3-dimensional triangle that has all these beveled angles. And I'm not going to bore you with the details of how you actually design these things in silico. And so instead what we'll look at is we'll just look at how these structures are made. So here we can see a TEM image of some triangular subunits. And it seems like we can make these nice kind of DNA origami structures. But we can do a little bit better than this because it's hard to see if we encoded all the right information into this type of object. And so we can also do something called cryo-electron microscopy to see the 3D shape of the structure that we've done. And so here's a 3D revolution. Hopefully this movie's playing. Yeah, a 3D revolution of this object. And what you can see are these kind of regions of cylinders here. These are these double helices of DNA. And if I just play it one more time, maybe. Yep. You can also see that these edges are a little bit tilted, right? So it seems like we're able to design in this type of binding angle into our object. So let's see how this type of subunit does in terms of assembly. And I should mention one more thing. That kind of rough object just looked kind of flat and uninteractive. But what we can do is we can also pull out single-stranded bits of DNA from all of the sides. And if we encode the right sequences there, we can make sure that we get the right binding between sides. So white binds to white and blue binds to blue and so forth. And so let's see how this object looks when we try and assemble it. And so DNA is nice in the sense that it's very temperature sensitive. And so you can really get a whole range of binding and energies just by changing the temperature of your assembly. And so if you mix these particles together, you put them at high temperature, you just get monomers. If you have them at too low of a temperature, they'll start to bind, but they don't have any off rate. And so they tend to make these kinds of elongated, kind of holy things. You can see hints of it being filamentous, but it doesn't make a tubule that we want. But if you tune the binding energy just right, you change it to the right temperature, you get these nice tubule structures. So this seems great. It seems like we were able to design this type of self-assembly particle to make a cylindrical sheet. But if we look around these kind of TEM grids, we don't just see one type of tubule. So this was a little bit of a bummer. First, we thought we did a really good job of defining angles, but then when we look, we get a distribution of different types of shapes. So here are three transmission electron microscopy images of different assemblies that we find. And the first thing that pops out to me is that they have different diameters, right? So they have different widths. But if you look closely, you can also see that the patterning on these different images is different. And this is because when you make these kind of samples in TEM, our structures get flattened. And so you have two superimposed triangular lattices on top of each other. And when you look at them in transmission, if they have different angles between them, you'll get these types of moire patterns showing up. And so this tells us that we also have different helicities in here. And so we can quantify the kind of polymorphism that we're seeing in these tubule distributions by looking at a distribution plot like this. So this is a plot that shows you the relative frequency of different types of tubules. Just as a reminder, we had these m and n numbers. So each point on here corresponds to a different tubule. These are the points for these specific types of tubules. And the relative size of these points is just the relative probability of getting these different states. So you mostly get stuff up here, but you can get some out here, right? And so this seems like an issue. Even though we try and encode a particular geometry into our subunit through beveling these angles, we still assemble this kind of broad distribution of states. And so we want to think about how can we try and tame this polymorphism that we see in our assembly. And to do that, we need to think about why are we getting this? So here's a cartoon that I like to use to illustrate this type of problem in self-assembly. So if you just have two little bricks that have a designed target angle, right? You can design this angle so that hopefully when they come together, they form a ring of 10. But because our subunits are really small, they're kind of soft, thermal fluctuations will give rise to some fluctuation around this angle. And so you can easily close it at some neighboring state, say a 9 or an 11, right? And so we need a way to try and get rid of these nearby accessible states. And one way that we can try and do this is we can go back to this picture of different pathways in assembly, and we can try and borrow something from this idea of addressable assembly, right? If we're able to add more complexity into this system, maybe we can try and improve the specificity of this structure. And so in this ring example, what that could be is that instead of just a single type of subunit, I now have a red and a blue wedge, and we can encode the interaction so that red binds to blue, but not to itself. Then we can see that for these odd numbered ring states, you would have to have a red-red bond or a blue-blue bond, right? And so if you can effectively introduce this type of interaction specificity, you can prune away these nearby states. And we can do the same type of, we can play the same type of game with our tubule, right? So here's two tubules that are very similar states, a 10-0 and a 10-1. And again, we can go in with our paintbrush and we can paint these triangles and make rings of blue and red. And what we see is that if we do this type of painting, if we follow the same painting rules, on this cylinder, what we find is that for this 10-1 state, you would have to have this seam running down the side where you would have this kind of lateral red-blue binding, which wasn't allowed in our original coloring up here. And so the effect of adding colors in this tubule type system is that for this particular coloring, you would disallow these odd numbered end states, right? And so adding complexity into the system is the equivalent of kind of reducing the density of states that are allowed to assemble because you have this additional complexity. And this type of additional coloring is pretty easy to add into our experimental platform, which is nice. So here's an example of a tubule. And remember that we had these kind of base monomers that have single-stranded DNA that we pull out from the side, where if you just specify the sequence, you can specify the binding. And so what we can do is we can use 3 copies of the same base subunit, but just change the specificity of the sticky ends that are being extruded from the sides. And so by looking at this cartoon of the tubule on the left, we can try and infer what binding rules we would need to use. And so we would see that red binds kind of in this vertical direction to itself, and then to the right it binds to blue, and the left it binds to yellow. And if we just look at these kinds of neighboring particles in this colored tubule, we can infer what interactions we would need to encode. And so this raises a question, right? If we can now add this type of complexity into our system, there's a question of like how much complexity do you actually need to add, right? We saw that one component wasn't enough. You get this broad distribution of states. There's a cost to having too much, right? If I make 100 different subunits, we're going to have some really angry grad students that are purifying all of these things. And so you want to have a nice kind of boundary between these. And so we're going to do this in an experiment. We're just going to make tubules with a bunch of different numbers of subunits and see how the distributions change, if they actually get reduced in the way we expect. So just as a reminder, here's our distribution for our one component tubule. Has this broad spread. This is an example of the structure that's forming on the left. And what we're going to do is we're going to incrementally add components to the system. So here's an example of a distribution of states with 4 components. And what we see is there's 2 major features here. One is that indeed we've pruned away the density of states that can form, right? You can see there's far fewer dots and the space between them has grown. But we've also successfully funneled probability into a fewer number of states, right? The largest circle here is bigger than the largest circle for the one component system. And we can continue to do this now with a 16 component assembly. And we see that we basically get all of the assembly funneled into a single state. So this seems pretty good. And if we look at the probability of getting these different states, we get a really drastic increase in yield. On the left, for a single component system, the most probable tubule state had 13% of the structures, but we get up to almost 94% on the right? So we've almost fully specified the types of assembly that we get by adding this complexity. Okay. And so we can look at how the kind of yield of this most populated state scales with the number of components that we add in our system, calling this our number of colors. We see basically kind of a linear trend, and then it starts to plateau when we get to larger numbers of components. And so it'd be nice to know kind of what physical parameters are driving this. And one obvious example of this is if thinking about the ring again, right? You could imagine that if I have fewer components in a ring, the kind of cooperative fluctuations of smaller numbers of things would be less than larger numbers of things. And so we want to see how does this change with the kind of size of our structure. So we go back and we design a new monomer that targets a smaller diameter of tubule. Here's an example of this one and its distribution. Just as a reminder, the previous structure we showed you, it was sitting up here. So this is just a kind of a narrower tubule. And again, we can play this game of coloring in this structure, adding more complexity, and we can again see this kind of increase in the probability of a specific state for the system, right? We go from here having some probability and funneling it almost all into this 7-0 state. And so we can now plot the yield, the maximal yield, as a function of the complexity of these narrow and wide tubule case, and we see the same general trend. But now we want to, everyone loves scaling things together because it's very informative about the underlying physics. And so we want to have some meaningful scaling between these two things. And so it's useful to go back and think about what's going on here, right? And so the way we like to think about how fluctuations are arising in this system and competing with this complexity is by looking at graphics like this. So here's our example tubule. And if we think about on our triangulated sheet, we have our target state. Then what we can imagine is that there's kind of an area around this target state that's accessible to these thermal fluctuations. And what's happening is that as we're adding colors, you're basically adding another length scale into this problem, which is the spacing between similar vertices in this colored system, right? So now in this three color plot, now there's this distance between similar vertices here. And what that does is it removes some of the states from inside this area accessible by fluctuations. And if we keep increasing our complexity, we can eventually make it so that there are no nearby states within this accessible area. And so we can think about how these two length scales grow, how this area of fluctuations grows, and how this length scale from the coloring grows. And so to think about this area of fluctuations, we just like to think about kind of the elastic energy for a membrane. So if you just have some sheet of components, it has some bending modulus, has some area, and it has some preferred curvature, then we would expect that if that kind of deviations from this preferred curvature are harmonic, then this tells you that kind of from thermal energy, you would expect a kind of fluctuations of the circumference of this cylinder to go like the target cylinder diameter divided by the square root of the bending rigidity. And so that means that the area of this type of accessible region of fluctuations should go like the circumference squared divided by the bending rigidity of the sheet. This then we can compare to the separation between similar vertices. This is something where this should grow like the square root of the number of colors. And if we compare these two length scales, we get that we should have some crossover between these two things happening when the number of colors is roughly the circumference squared divided by the bending rigidity. And so now we can go back and we can try and scale this plot of the specificity of our tubule assemblies. And what we can do is from our single component case, we can get some estimate for the bending rigidity. This and some other measurements, we have a value of around 7 kT. And if we plug these things in, we get this really nice scaling of the specificity, right? And so this is great. This means that now in these types of tubule assemblies, we can really drive the assembly of just a single type of target. And so if we go back to kind of initial goals, the first question we want to ask was, can we design and assemble these things? And the answer is we certainly can. That's really nice. And now that we have this toolbox built up of putting in curved subunits and adding specificity to the system, we want to ask what kind of progress can we make in targeting these types of shapes. And so what we're going to think about is that this is a bit trickier, right? Because along this shape, you have regions of positive curvature and you have regions of negative curvature, right? And what we know how to do is we know how to make a cylinder. And so the question is, can you do something to this triangulated cylinder to somehow put a bend into it? And so this is a project that was mostly done by Mason Price and Daichi Hayakawa. Mason Price is this phenomenal undergraduate student we have here at Brandeis who really drove this work forward. And what they were thinking about was they wanted to think about this idea of kirigami, right? Kirigami is this technique where you take paper and you cut holes in it. And when you stitch these holes closed, you can induce out-of-plane bending in this sheet. And so the question was, can we use this type of technique now on our cylindrical surfaces? And so the idea is if we can translate this over to our triangular system, right? There's two things you can do. One is you can try and cut out material, right? So if you make a cut and you close this up, you're introducing an angle deficit into this region of the triangulated surface. And what that does is it adds a region of positive Gaussian curvature. But another thing you can do is you can join regions that have that have a few triangles in them. So here we have this fourfold vertex and this other fourfold vertex, right? They've already had some material cut out, but if you join these two vertices together, you'll now have a region where you have an angle surplus, and this generally adds a region of negative Gaussian curvature. So the question is, how can we try and add these two features into this kind of cylindrical geometry we've been discussing? And so what we'll do is we'll start with a cylinder. You have your triangulated sheet, you roll it up, and then what we're going to do is we're just going to cut a hole out of it and try and stitch it back together. And what this does is that at the corners of this hole, you'll have these regions where you have angle deficit, and in the middle here, you'll have angle surplus. So hopefully in your closed structure, you'll have regions of positive and negative Gaussian curvature, which is just what we needed to make this kind of bent tubule. But one thing that isn't obvious from the get-go is how, when I stitch this thing together, how this thing will actually close on itself. And so one tool that we really like to use in our lab when thinking about equilateral triangles are these nice snap triangles. If you haven't seen these before, I highly recommend buying some and playing around with them. It's a really nice tool for just making different types of surfaces. And so we can snap these things together to form a cylinder that we then remove a patch from. And then we can try and close it in the way I showed before. And what you find is that it makes this type of shape. It has these two tubular sections at the end, and then it has this little junction region. And it seems like it makes a 90-degree bend. So that's nice. This is nice suggestive visual evidence that this thing will close into this type of shape here. And we can then ask, how can you kind of use this motif of a bent triangle to make more interesting structures? And so I can take two of these kind of bent junction regions, and you can start sticking them together in different ways. You can either make them so that they snake in the same plane, or they start to wrap around on themselves. This one on the right is you have two wedges that are not in the same plane, so this is pointed out towards you. And so it seems like by combining these things together, these wedges and these rotations between adjacent wedges, you can try and get access to these different types of morphologies. And so let's make this a bit more concrete so that we can play around with it. Because once you know that these triangles can make these kinds of interesting shapes, we now need to know how to encode this into some kind of assembly system. And so this is the scheme that Mason came up with. You have this triangulated sheet, and you have these different regions that we've painted in blue and red, and these holes. And so the first thing we do is we specify the length of this blue region. This will eventually make the kind of tubule in our structure. So we'll denote this, the edge length here as T. You can also specify the length of this region as L. There's the size of this kind of junction region, which we call D. And then there's this R region, which is if we now have this one hole and we want to have multiple bends in our tubule, we want to know we need to know the relative phase between holes along the length of our cylinder. And so there's this parameter R, which just has to do with a kind of relative placement of a hole in the next layer. And so these things together give you these four parameters for defining the way that we can take a cylinder and cut these diamond-shaped holes out of it. Just to give a bit more intuition for what this R parameter does is you're kind of shifting this relative location of the next junction, which corresponds to just a rotation around the circumference. And if we think about this kind of closed state, it's just a relative twist of the next tubule section. And so this is what we're going to try and do and see if we can make some cool larger scale structures with this. And so the first step to figuring out how to make this realized in the assembly system is that because every region of this structure has a different local geometry, right? It's either part of this positive Gaussian curvature thing or this negative Gaussian curvature, or it's part of the tube. We need to know how to encode these local regions of curvature. And so we first take this triangulated tiling, and then we're going to add some color to it. So we can use the tools we had from before where we are able to color our cylinders in different colors. And so we just do the same thing here, except that we just leave out the whole regions. And then we're going to add in this stitching rule, right? So the two things we need to know for self-assembly two of the things we need to know is we need to know the angles of binding, and we also need to know the interactions between them. And so between this coloring and the stitching rule, we can infer all of the local interactions that you need to program into this type of system. The next question is, how do you get the local binding rules between them? And so we can take this initial set of interactions, and we don't really know a priori what the geometry of this structure is going to be. And so we have to simulate, or what we do is we simulate it. I don't know if you have to simulate it. That's a good follow-up question for discussion. But we simulate the structure. So we start off by having this tubule with parts cut out of it. And then we slowly just try and relax these bonds together and enforce these closings. And it seems like for this particular starting condition, this thing likes to fold up into a toroid, which is neat. So we found a way to make this kind of closed toroidal structure. And once you're in this final configuration, now we can go in and we can measure the relative binding angles between triangular faces. So each edge now we can correspond to a binding angle. And so now we have the things we need to make a self-assembly system. And if we can also look at these other values of this R parameter. So using the same kind of base triangulation, but now we just raster this whole, we also find that this scheme allows us to make either left or right-handed, right or left-handed helical tubules, and also this cool serpentine structure, which just oscillates back and forth in plane. And using the kind of DNA origami system that we had set up before, we can actually go and realize these things in the lab. And so Mason had this heroic experimental effort where he synthesized all of these different components. He was able to encode all of these different angles, and you can really get these you can make these exotic curved surfaces now in experiment, which we think is really exciting. Okay, and I think I'll end the, oh, and I just want to mention one more thing. So those are just 4 examples of different structures, but using this kind of TLDR scheme that oh, sorry, I just saw a chat comment. Using this TLDR scheme, you can get a wide variety of different types of toroids and different types of helical shapes, and we're excited to see where this thing goes. And so hopefully, yeah, hopefully I inspired some thought for you. That is a cool self-assembly playground. And when you use triangles, you can make really interesting shapes. I showed you how coloring these types of cylinders can give rise to really high specificity structures. And if you also consider this kind of kirigami-inspired method, you can start to make these interesting varying curvature shapes. But there's a lot of questions we still have open to this. On the colored tubule side, as an experimentalist, when I see a scaling that goes like the size of your thing squared, right? We had that this kind of ideal number of colors goes like the circumference squared. That's a little daunting, right? Because if I wanted to make larger and larger shapes, that's going to be really costly in terms of complexity. And so we're always interested in thinking about, are there more economical ways that you can reduce the polymorphism in these types of assemblies? In terms of this kirigami method, we showed you this one diamond hole, but it's interesting to think about, in general, what types of shapes of holes are allowed, which ones can you actually stitch together that form a non-intersecting geometry, and what happens if you start to do this kind of cutting and stitching method on not just these cylinders, but on different types of surfaces. Is that viable? Does it work? We're not sure. But with that, I'd like to thank you for your attention, and I'm happy to take questions.